Hello everyone, and welcome to today's webinar in the SERDP and ESTCP webinar series. My name is Rula Deeb. I'm a principal at Geosyntec Consultants in Oakland and the organizer of the webinar series on behalf of SERDP and ESTCP. I will be facilitating today's call. The webinar will consist of a brief overview of SERDP and ESTCP by Dr. Andrea Leeson, followed by a sneak peek at a few of the upcoming webinars in this webinar series. Following Andrea's opening remarks, we will transition to the technical portion of the webinar. Today's event features two speakers who will discuss a quantitative framework and a management expectation tool for the selection of bioremediation approaches at chlorinated solvent sites. First, Ms. Carmen Lebron will introduce the topic and will provide the project objectives and technical approach, followed by a short Q&A session. She will be followed by Dr. John Wilson, who will detail the results of this project and provide some case studies. We will conclude the webinar with an interactive Q&A session, including both our speakers. Today's broadcast will be listen only. You may submit questions by using the chat box on the left-hand portion of your screen. You do not need to wait until the Q&A period to submit your questions. In fact, we do encourage you to submit questions in advance of the Q&A sessions. With over 450 attendees on today's call, it is logistically challenging to open all the lines for all our questions. Therefore, the phone lines will remain listen only throughout the presentation. With that, I would like to introduce Dr. Andrea Leeson. Andrea is the Deputy Director of SERDP and ESTCP, as well as the Program Manager for Environmental Restoration. Andrea has been with SERDP and ESTCP since 2001, and before that, she was a research scientist at Battelle Memorial Institute, where she conducted research on in situ bioremediation. All right, Andrea, the floor is yours.
Thank you, Rula. I'm happy to welcome everyone today to SERDP and ESTCP's webinar. Before we start the webinar, I'd like to tell you a little bit about our programs. SERDP is the Strategic Environmental Research and Development Program established in 1991 by Congress as a partnership between the DoD, the Department of Energy, and the EPA. SERDP's mission is to identify and address high-priority environmental science and technology opportunities that focus on the DoD requirements. SERDP funds both fundamental research as well as advanced technology development that ultimately impacts real-world environmental management. ESTCP is the Environmental Security Technology Certification Program in which we demonstrate innovative environmental and energy technologies. These investments capitalize on past investments under SERDP or other research programs and are designed to transition technologies out of the lab and into the field. Especially important in all ESTCP demonstrations is the ultimate transition to implementation and regulatory acceptance. SERDP and ESTCP are complementary programs with much of SERDP research occurring at the lab and pilot scale with occasional field efforts, while ESTCP demonstrations are primarily at the pilot and field scale, although occasionally supporting lab efforts are conducted. We have 4 program areas in SERDP, 5 in ESTCP. The Energy and Water Program area is only in ESTCP, while the other 4, Environmental Restoration, Munitions Response, Resource Conservation and Climate Change, and Weapons System and Platforms, are SERDP and ESTCP programs that are managed jointly by a specific program manager. Our webinar today is focusing on research and demonstrations that were conducted under the Environmental Restoration Program area. Environmental restoration has essentially 5 main areas of research: contaminated groundwater, contaminants on DoD testing and training ranges, contaminated sediments, wastewater treatment, and risk assessment. And of course, as you can see, these are very broad program areas, but we truly try to cover a very wide range of issues associated with these 5 areas. Our webinar series will be highlighting research and demonstration efforts from each of the 5 program areas. Shown here are some upcoming webinars, and as you can see, the webinars cover a broad range of topics. with upcoming topics covering innovative tools for species inventory and monitoring, to blast noise measurements, and to munitions mobility. The next webinar focused on research and demonstrations that were conducted under the Environmental Restoration Program area will be on May 28th and will cover the management of munitions constituents on training ranges. You can find more information about upcoming webinars on the SERDP and ESTCP website. Registration is now live for the March 26th webinar on innovative tools for species inventory, monitoring, and management. And I hope you enjoy the webinar today. Rula, I'll turn it back to you.
Thank you, Andrea. And we're going to go ahead and get started with the technical presentations. Our first speaker will be Ms. Carmen Lebron. Carmen is a private consultant with 33 years of experience in R&D dealing with remediation of both fuel and chlorinated hydrocarbons. Prior to becoming a private consultant, Carmen was a senior environmental engineer for the Navy in Port Hueneme, California. During her tenure at the Navy, Carmen managed over 14 SERDP and ESTCP projects on remediation of chlorinated ethenes, use of molecular biological tools as diagnostic tools, and monitored natural attenuation. And with that, I'll turn it over to you, Carmen.
Thanks so much, Rula. This morning, as Rula mentioned, I will be speaking about the background of this project, and then John will come in and talk about actually the framework application, which includes the review of regulatory requirements, the intended application of the framework, and the decision logic behind the framework. He'll also present 3 completely different case studies which will showcase the different application that the framework has. I'll also like to introduce other members of this project. Todd Wiedemeyer with Wiedemeyer & Associates. He was the one who developed the original M&A protocol, and this project is largely based on that protocol. We also have Dr. Frank Loeffler, a world-renowned microbiologist who brought to this project his intricate knowledge of biological processes. Yi Yang is a graduate student with Frank, and he was the one who developed our BioPIC, or our screening tool. Mike Singletary with NAFAC Southeast, whose site manager paradigm and needs were kept in mind throughout the entire project. And last but not least, Dr. Rob Hinchy with Integrated Science and Technology. Rob is also a world-renowned PI who participated in the development of the original protocol over 18 years ago. The technical goals of this project were, in essence, to update the M&A protocol, which was published back in 1998. Specifically, we wanted to incorporate the new science that has been developed since close to 20 years ago, back in 1996 when Todd first wrote the initial protocol. We also wanted to integrate this decision framework into an easy-to-use application. And as you recall, the original protocol was simply a sheet where all these parameters were listed, and you place measurements of those parameters at your specific site into that sheet. So now that we're in the 21st century, we thought that could use an update. By virtue of this, we also wanted to create a paradigm that would make it easier to users to select between M&A, biostimulation, and bioaugmentation. We carried out this project in 5 tasks. The first task was to identify the parameters that were of interest, and the criteria that we used about the impact was the impact on the rate of attenuation. This was something that John suggested early on in the project, and it made sense because the definition is consistent with the OSWR directive. We then down-selected those parameters to a manageable size since we knew that for each one of those parameters, we were going to have to determine the impact on the rate of attenuation. Following that, we developed the framework logic, which are the charts and flow diagrams on how site managers should address the selection of a bio approach. And after that, we developed the tool, the screening tool that users will use to apply this paradigm. The pathways that are addressed in this framework include complete reductive dechlorination, partial reductive dechlorination, also known as DCE accumulation, aerobic biological oxidation, and abiotic processes. We didn't address these pathways because one was more important than the other, but mostly because at one site users may have more than one of these conditions. And for comparison purposes, the 1998 protocol only dealt with reductive dechlorination. So let's go on and look at each one of these tasks in a bit of detail. The first task was to identify the parameters. And we started out with those 27 parameters that were spelled out in the 1998 protocol. During that parameter ID task, we also wanted to focus on parameters that were applicable to most pathways addressed in the framework. So after several calls and meetings, some clear winners were emerging. And at that point, we had reduced the 27 parameters to 11. Now, consistent with the 98 protocol, in that list, we have the concentrations of the VOCs, oxygen, pH, and ethane. And we also had others that were important in biological reductive dechlorination, but also abiotic dechlorination, like Fe2 and the sulfides. In this list, we also consider some new parameters, as in our collective experience, we thought or believed that these may impact the attenuation rate. So these new ones included the Dehalococcoides density, that is the ratio of Dehalococcoides to total bacteria. We also had the ratio of the reductases to Dehalococcoides, and we have biologically available organic carbon, or BOC, magnetic susceptibility, and acid volatile sulfide. Once we got into the task of down selecting the parameters, we emerged with a list of selected parameters that we could use to further evaluate our assessment on impact of those parameters on the rate. Our collective experience and many references led us to this point. Some of these parameters are routinely sampled for, thanks to the original 98 protocol, but some were new. And we still made a push for those because there was preliminary evidence that they could impact the rate. And those are the ones listed at the bottom of this view graph. During task 3, which is essentially what ended up taking the majority of the time of the project, we were to estimate the parameter impact on the rate. So through this exercise, we reviewed data from real sites to determine the relationship between the parameter and the rate of attenuation. We started out by sending several emails asking for data, and I want to thank those of you guys who participated in this exercise and provided us site data. Paramount to this exercise, we also had what we call poster child sites. And these are sites that are well published, for which there's notorious amounts of data. And the pathways in those sites have been identified. In our case, we benefited from John, Mike, Todd's and Rob's intricate knowledge of those sites. I've listed those poster child sites, and many of you may recognize those. Our initial goal was to come up with data from 10 sites, so we had a statistically significant population of sites. Thanks to all of you guys, we ended up with 65 sites and over 3,900 wells. During this exercise, the question to answer was, how do different values of each one of these parameters affect the rate constant? So when it was time to estimate that impact, we followed a methodology that had been used and validated by John Wilson and others. We followed, this was published in 2006, and in this 2006 exercise, the purpose was to establish or determine a valid correlation between Dehalococcoides densities and the observed reductive dechlorination rates at 6 sites. After that, they used Spearman correlations to analyze these relationships. And the conclusion of that study was that useful rates, which are typically higher than 0.3 per year if the remediation is required in 30 years, were observed for DCE and vinyl chloride when Dehalococcoides densities exceeded 10 to the 7th. So we used that same methodology, and let's move on to the next, actually. I'm showing in this view graph number 26 the same chart that was used in 2006, only now at this point we have added the new sites. And the new sites, I mean the ones that resulted from the call from the emails that I sent out. So let's look at one of these distribution plots after we received the new data. And in this one, in 26, we are comparing Dehalococcoides densities to attenuation constants for cis-DCE. Again, each one of these points is a rate constant that has been estimated, and it has been measured against the concentration of Dehalococcoides. What we did is we drew a line extending from 10 to the 5 to 10 to the 10th Dehalococcoides gene copies per liter to include the points that we can reasonably attribute to the action of Dehalococcoides densities. The line was drawn using professional judgment because A, we know from the literature that those are efficient densities, but also because we can see on the y-axis that it produced or achieved acceptable rate constants. So we then drew another line with the same slope encompassing the attained rates that we knew are acceptable for the site, and we grouped all of these in a box that we called sites that we can reasonably assume that the Dehalococcoides densities are responsible for the rate. We did this analysis for all of these parameters. We related the rates of attenuation of the individual VOCs to Dehalococcoides, Dehalococcoides densities, etc., etc. And we produced numerous distribution charts. At the end of that exercise, there were only a few of these parameters that we realized really impacted the attenuation rate. And those parameters are the Dehalococcoides density, although not in the case of PCE, as there's other microbes degrading. It was also directly related to magnetic susceptibility, to ferrous sulfide, methane, and ferrous iron. So we use these parameters to design and develop the decision framework logic, which is in this next few graphs. I don't expect you to be able to see the entire flow chart, but the rationale behind the process, again, is the OSWR directive for M&A. And I want to point out a few of these elements in that flow chart. So in more detail, here how it all starts. You first have to estimate the rate constant for the transect of the well of concern. And then at that point, you're answering, does the rate of attenuation meet the goal established by the regulators? If the answer is no, you have two options, either biostimulate if the reductases are present, or biotic and biostimulate if they're not. Let me point out that we recognize that reductive dechlorination cases are a no-brainer. So you see it. By that I mean that you see a reductive chlorination case and you immediately recognize it. But where this framework logic is extremely useful is in deciphering what's going on at the class 3 sites that were mentioned in the original protocol. So on the other side of the yellow rectangle where it starts is where the challenge arises. If natural attenuation meets the goal, the only other question you need to answer is if there's a second line of evidence required. If there is, the flowchart takes you contaminant by contaminant through a rationale encompassing the different potential pathways that could be important. You may also notice different numbers under each one of these boxes, and those correspond to different help boxes that are also available through the program BioPIC. This is just a point in the flow chart where we encounter the biological component. But what I also want to point out here is that this application allows users to enter their very own data. In this case, we've gotten to the point in the flowchart where the user needs to address whether it's magnetic susceptibility. The minute the user clicks on that on this point, then the program will take them to a spreadsheet titled Magnetic Susceptibility where they can enter more data. The last task was to develop all of this paradigm into an easy-to-use application. So we ended up calling that application BioPIC, which stands for Pathway Identification Criteria. And in other words, it tells you which path to go. It essentially guides you through the entire flow chart, and it's an Excel-based spreadsheet with macros where you input your own parameters. So I'll go quickly through an example because I know John will talk more about this later. In this case, all these points where you see input, that's the data that you're going to be inputting. So you put in your different rate constants for your different contaminants and your qPCR numbers. And what BioPIC does is it goes into our distribution plot and it locates your very own number. And at that point, it assumes that if it's within the blue box, it could be reasonably attributed, in this case, for example, to the Dehalococcoides densities. The reason why this is important, I like to think, it's threefold. First, It saves us money in that the characterization at a site is more tailored towards the predominant detoxification pathway. Also, the framework follows exactly what EPA prescribed. It guides users in the most appropriate bioremediation approach, and bioremediation has been reported to be one of the most cost-effective remedial approaches. And many of you have seen this chart. It comes from a 2013 Defense Environmental Restoration Program annual report to Congress, and it's obvious that the annual budget for the restoration will never match the cost to complete. Therefore, we must do more with less. As Rula had pointed out in the beginning of the presentation, I've had several ESTCP projects, and in the past 22 years managing this many projects, I've never encountered a project where at the end of it, everything is in place for it to happen. The regulatory infrastructure is in place, the technology is in place, and the framework is in place. And with that, I think we can open it to questions.
Yes, thank you, Carmen. I would like to remind our audience to submit questions via the chat box, which is on the left-hand portion of your screen. Carmen, we've received a number of questions, and the first one has to do with feedback from the regulatory community regarding this framework. Have you received any feedback? Are there going to be efforts planned to do outreach to regulators to inform them of the technical details of this framework?
I will begin that one, and I will lead that one to John. We have plans on going out to both different RPMs, DoD RPMs, and EPA RPMs, hopefully in a wide training session, a few hundred people, hopefully. But John is the one that has been preaching, I don't want to call it preaching, but spreading the word to the regulators, and he's done several presentations. John, do you care to jump in on that one?
Well, it's just that we continue to do what we've been doing, and we look for further opportunities to expand our outreach. At this point, best to coordinate that through Andrea and SERDP STCP. But we're willing to bring more details to anybody that's willing to listen to us talk about it, and particularly with people who are willing to share with us new information and new sites that would expand our understanding.
All right, thank you both. Carmen, if one is limited in the number of parameters that can be sampled due to budget or other constraints, which three would you recommend analyzing for?
Well, we've gone through this exercise in the past week a few times, and granted, there's a few differences amongst the three of us. I would suggest Fe2, Dehalococcoides density, methane, and if you see that your site is following an abiotic pathway, definitely magnetic susceptibility.
Thank you, Carmen. Several people have asked for you to please elaborate on what you mean by magnetic susceptibility.
Let me.
That's John. Perfect.
Okay, John.
It'll move it up. I was going to talk about that later in the presentation. It means exactly what it says. An iron nail is not a magnet. But if you hold a magnet next to the iron nail starts to act like a magnet. It's susceptible to a magnetic field, and it propagates a magnetic field. So magnetic susceptibility is a quantitative estimate of this tendency to propagate a magnetic field. It's acceptable to the field, and then it becomes part of the field and extends the field.
Thank you, John. Are you going to talk about how you can make these measurements later on in your presentation?
Yes, I will.
Excellent. All right, Carmen, question for you on slide number 26, and I'll take you back there on the screen. How do you explain the high rate constants for DCE on the slide at low DHC densities?
If you don't mind, let me do that.
Yes, please. I think those were the sites that were biostimulated.
Yes, and we're looking at it. If you look at that, you actually see two populations of data points. One that has a trend that goes from higher rate constants along with higher densities of the organisms, and then another trend that just basically ignores the DHC and is independent of DHC. Many, if not all, of these points are from stimulated bioremediation projects. And I think, I don't know, but I think that we're looking at the influence of abiotic degradation, perhaps by green rust, which are hard to measure, are ephemeral, but are very reactive against chlorinated solvents. And so I think what we're looking at here is the additional, and in this case, overriding degradation of DHC by abiotic processes. And because the abiotic process is controlling the rate, then it's not related in any obvious fashion to the density of the Dehalococcoides organisms.
Thanks, John.
Excellent. One last question for Carmen before we move on to the results and case studies. You talked a little bit about bioavailable organic carbon measurements, but you didn't include that in the list of parameters. Why is that, Carmen?
We had several issues with BOC, and it's not related to the test itself. We think that the test itself is very robust and is actually one of the critical tests to be done if you are trying to make a case for M&A, because it does offer a fuzzy warm feeling and comfort about the sustainability of M&A. Unfortunately, it's a test for which you need to pull out a borehole. So it's not being done a whole lot. And when we started looking for site data for it to correlate it to the rate of attenuation, the data was just not there because very few sites do this kind of analysis.
All right. Thank you very much, Carmen. And with that, we're going to transition to the second portion of the presentation. We've received a lot of questions that we haven't answered yet, but we will get to them after John goes through some of the results and conclusions. So our second speaker is Dr. John Wilson, who really needs no introduction. John has extensive experience in natural attenuation processes and in bioremediation. He is currently a principal scientist with Scissortail Environmental Solutions. Before that, he served as a research microbiologist for USEPA at the Kerr Environmental Research Center in Ada, Oklahoma for 35 years. John's research at EPA focused on natural attenuation of BTEX compounds, fuel additives, and chlorinated solvents. He also did a lot of research on in situ bioremediation of chlorinated solvents. So with that, I'd like to turn it over to you, John. Thank you so much.
Well, thank you, Rula. I'm going to start by talking a little bit about the regulatory guidance that frames our approach and appropriate use of natural attenuation. This simply is the citation to what we call the OSWR 1998 guidance on the use of monitored natural attenuation in the federally regulated enforcement actions. The following slides are actually quotes from the guidance. And sometimes I wonder if people actually read all of it or if they just read summaries. But from the very beginning, the two very important points came out of EPA's stated position on M&A. It's built around a comprehensive and detailed site conceptual model, and you use that site conceptual model then as a framework for evaluating the efficacy of all of the remedial alternatives, not just M&A. But for M&A, like any of the other alternatives, you have to understand the rate of the processes that are bringing about the remedy. And if you're in a Superfund regulatory environment, you need to understand how those rates are related to achieving the goals within the anticipated time frame that's required by the ROD. The evidence to support M&A as a remedy is organized in tiers. The first one, of course, is the historical groundwater monitoring data. And so we built our framework around organizing and understanding that historical monitoring data to recognize a clear and meaningful trend of decreasing concentration over time. And then if the first tier of evidence indicates that monitored natural attenuation may be appropriate, that can be supported by the second line of evidence, or second tier, which are geologic and geochemical data that can be used to understand the mechanism of attenuation and the rate at which the processes that are carried out will reduce the contaminant concentrations. And so basically what we did in our ESTCP framework is put some how-to-do-its around the OSWR 1998 guidance. Now, there's a caveat here about the guidance, about our framework. It's not a one-size-fits-all. It really was designed to work with M&A that's in what I would call the RCRA framework, where you have groundwater contamination on your facility and you're required to contain it on your facility and not let it be spread to share with the neighbors. And so the regulatory question in that particular context is, well, will the plume impact a receptor? And that's defined by the regulators. And then we turn that will it impact a receptor into an engineering question. Is the rate of attenuation going to bring the highest concentrations on site to acceptable concentrations before it gets to the receptor? And that's done using a computer model to extract rate constants from the field data to see what rates would be necessary to make M&A protective. And then the next step within our framework is to see if we can find other lines of evidence that support and defend those rates that are extracted from the monitoring data. So all of this logic and process is incorporated into a framework called BioPIC. Basically, it's a little expert system. And we're going to work through that for examples in a few minutes. But it's important to offer up a caveat about what it doesn't do. It doesn't tell you in the Superfund context when you're finished. In Superfund, if the entire plume is required to meet the goal, then that performance depends on your your successes for source control and on the kinetics of natural attenuation of the sources. And the kind of information that goes into BioPIC cannot be used to understand or evaluate the sources. BioPIC is designed for and uses information from assessment processes that understand the rate of biodegradation of the contaminants once they're in the groundwater. So I guess in a way to summarize, the framework in BioPIC are best applied to a RCRA-type context, and there are other guidance and processes that are suggested for understanding the Superfund question of when is it all clean. So with that limitation, or rather that focus in mind, let's take a look at the decision logic, and Carmen showed you this slide earlier. The point I want to make with this is each of those blue diamonds is associated with what I call a bright line. I have been disappointed in decision logic that's been produced by government in the past, where you have a very qualitative and a subjective decision point. Like, is M&A happening? Well, that all depends on who thinks what about it. Decision logic is useful really for someone that's smart but doesn't know much about the system and is trying to learn something fast. And if you're asking them to provide a lot of professional judgment, it makes the decision logic worthless. We created a logic that has the team's expert judgment embedded in each diamond. So for each diamond, we offer a clear distinction about whether it's yes or no. And BioPIC works you through those questions. And I'll show you in a little bit how you can determine if natural attenuation meets the goal. And if it does, you say yes. And then BioPIC brings up the second question, well, do you require a second line of evidence? And that, in fact, is determined by the regulator involved with the site. Let's say that regulator says yes. Then BioPIC walks you through the 4 chlorinated organic compounds that this framework was designed to describe, one at a time. Vinyl chloride first. The first question is asked, is it present at significant concentration, yes or no? If it is, then is it degrading? And so the point of it is the whole logic is reduced to a number of simple, discrete, answerable yes or no questions. And for each of those questions, the decision criteria are stated explicitly, and then there's help on how to get information if necessary to answer the question in the decision criteria. So I'd like to walk you through a number of case studies to illustrate this whole process. This first one is actually one of the best-known landfills and golf courses in the United States. It's right at the end of the runway of the Naval Air Station in San Diego. And it was a common practice 50 years ago to dispose of hazardous waste with the landfill, basically get out with a bulldozer and dig a trench and dispose of the materials as needed and cover it back up. And that was done. And if you'll look carefully at this slide, you'll see 2 little red marks, which were the bulldozed trenches that were covered back up, and then an oval shape that's enclosed in a blue line. And that's the distribution of known contamination that originated from that disposal practice. Now, to illustrate this process, we're going to have a regulatory boundary. Where do we care about the concentration? And for this particular site, that's not available, so I'm going to make one up. It's important to recognize that this is just for purpose of illustration. It has nothing to do with regulatory processes between the Navy and the state of California. But the state of California does identify the high tide line as identifying the boundaries of the waters of the state of California. So that red line then is the distance from the source of contamination to what would be conventionally described as waters of the state. And so our question is to determine if monitored natural attenuation is workable for this site, or whether it's needed at this site. Is in the absence of biodegradation, would we reach that receptor in excess of, and we use the MCL as a concentration-based standard. So the site is very well characterized. Each of those dots is a permanent monitoring well. You can see that there's a hot spot of chlorinated ethene concentrations that only extend a little over 200 feet from the source. So what we did with this is try to identify a plausible center line for the plume. And so I've plotted the logarithm of total chlorinated hydrocarbons on the y-axis and distance along the flow path on the x-axis. And for many plumes in this kind of plot, you get a straight line for the Plume Centerline. And so that blue line is my judgment that this should be what the Plume Centerline should be. And so that identifies 4 wells as being on the centerline, and then another 4 wells that are probably not. So I'm going to set up my flow model for the 4 wells that are along the Plume Centerline. We decided to use BioChlor really as a teaching instrument. At many sites you'll use a more complex model, a distributed parameter model, perhaps a 2-dimensional instead of a 3-dimensional model. But BioChlor is useful as a teaching tool because it encompasses all the processes and because it's easy to use, easy to learn to use, and basically because it's free. And so you don't need to see all the details on this. It just happens to be the BioChlor setup for this particular site. And to see if we need to have some degradation processes operating before this site would be safe, to make sure the concentrations didn't reach that receptor, you set the model up with 0 as the decay coefficient. And so no decay is happening. And we run the model, and it makes projections forward in time and space about the concentrations that would be accepted at that receptor at 1,500 feet from the source. And we can see that for DCE and vinyl chloride, the concentrations are orders of magnitude, many orders of magnitude above the MCL, which would be the standard for the concentrations that would be acceptable at the receptor. So if there's no degradation, if there's no natural degradation, the M&A doesn't work for this site. Then we went through a process of curve fitting. We basically added different values for the rate constants and ran the model until we got an acceptable fit to the field data. And so this is that best fit that I could get. Play around with different values for the rate constants until the projections of the model, which is the red line and the blue line, best fit the real data, which is the red triangles and the blue squares. And then the question is, are these concentrations below the standard before it gets to 1,500 feet? And at this site, clearly it does. So if we can satisfy ourselves that those rate constants are actually happening, that they're real, that they're justified, then monitored natural attenuation would be a very plausible alternative for remedy at this site. So the question then is, can we explain those removals? And what we're going to use is data on quantitative PCR assays for gene copies of basically a structural gene that's related to Dehalococcoides mccartyi. Dehalococcoides mccartyi is the only bacteria known that can degrade cis-DCE to vinyl chloride and then degrade vinyl chloride to ethene. So we're going to look for DNA associated with the bacteria that can carry out the process that we're interested in. And for the data set that we're looking at, the Navy did collect information on the density of that organism in 2 of the wells. One that's close to the source, and then one that's down gradient. The one that's close to the source has roughly 10-fold higher concentrations of the marker for the Dehalococcoides DNA. Now, what does this mean? Carmen showed you or referred to this particular paper earlier. We tried to do a similar thing 10 years ago, going around to various field sites where we had estimates on rate constants and looking at the density of the Dehalococcoides structural gene, and then plot the first order rate constant against the density. And then I show you this again to point out something. We compared the scatter or the cluster of, or the association of the rate constants and density to a rate constant and density for a pure culture of Dehalococcoides maintained in the laboratory. And so that's that black square at the end of the dotted line. But if you do a linear extrapolation of the pure culture strain down to the densities that are seen at field scale, then that also extrapolates the rate constants down to the rate constants that are seen at field scale. So we felt that that the DNA that we were seeing in Dehalococcoides at field scale was a reasonable and plausible predictor or reflection, probably reflection, of the rate constants for removal of those compounds at field scale. And so you've seen this plot before now several times, and again, that blue shape is professional judgment. Then just basically a bunch of us got together and said, well, now which of these would we plausibly think is the association of the rate constant with the density? So if you put in the numbers then, and the density at the well that's near the source, then what the spreadsheet does is it tells you what fraction of the rate constants in the benchmark data set predicted the same rate constant or a greater rate constant. Now for this particular site, those rate constants of 17 and 10 are very, very fast, 17 per year and 10 per year. And a half-life's on a scale of months or weeks. And so anybody that is looking at, I'll use the word more common sites, we get rate constants that are a tenth of that or maybe less. So the question is, well, is that really real that it can be that fast? Can we explain those huge rate constants from the Dehalococcoides cell density? And when we plot it up, and it's over there in the far right-hand corner, but it's in the blue shape. And we think that, yes, it is a plausible explanation for the rate of removal of cis-DCE at this site. And then that's the comparison for final chloride kinetics. And again, it's a corner of the blue shape, but it's in that range that our expert judgment said would be a plausible explanation for the rate constant. Let's take a look at the density data from the other well that was further down the flow path. In this particular case, 40% of the benchmark or poster child sites exceeded the rate that would be predicted from 3.47 times 10 to the 8 Dehalococcoides gene copies per liter. And that is again, it shows it where it plots, and it plots actually in the middle of the distribution for degradation rate constants for cis-DCE, and in the middle for the distribution for rate constants for vinyl chloride. Excuse me, let me go back and summarize this. Notice that the logic is backwards. The thing that people have been trying to do in the past is take a measurement of the organism and say, well, this organism causes this process. We should be able to predict the rate constant from the density of the organism. That has been a challenging and frustrating and disappointing activity. So the protocol turns it around. The framework says the first line of evidence is the field scale data. The first line of evidence is the actual rate constant that's required to explain the distribution in time and space. And the Dehalococcoides density now is a second line of evidence, and the question is, well, does it make sense? And so really all that's being done here is comparing this site, Site 5, North Island Naval Air Station, in October 2005, to the behavior of and performance of a large population of other sites, to see if its behavior is consistent with the behavior that was extracted for the other sites. We're going to take that logic now and apply it for some other processes. The framework actually has two abiotic processes. One is abiotic degradation on magnetite naturally occurring in the aquifer. The other is abiotic degradation on sulfide minerals that are produced in the aquifer through sulfate reduction. But we don't have enough time to talk about the sulfate reduction story. It would take another hour. I want you to know that it's in the framework, and if you're interested in abiotic transformations on sulfide minerals, you're welcome to look at it, and I'd be happy to discuss details with you if you have a site that's interesting. But I am going to talk about magnetite because it's simpler to see and it's much easier to explain. Many sediments in the United States have this mineral, magnetite. It's a mixed valence iron mineral. It's got some iron 2 and a lot of iron 3. And that iron 2 is chemically reactive. The iron 2 can chemically react with chlorinated solvents to degrade them. Most of the magnetite I think we're going to see is stuff that naturally weathered out of igneous and metamorphic rock, but it can also be produced in situ as a process or product of iron-reducing bacteria. We know that magnetite can degrade these chlorinated materials to oxidized products under either aerobic or anaerobic conditions. And when it does degrade it, it doesn't go through vinyl chloride. We have a fair number of sites in the United States that looks like what's been called cis stall. TCE goes to cis, and then cis just doesn't seem to go to anything. But if you model it up, there's a sink for cis. Something has to be removing the cis. At many of these sites, the water is aerobic, and so there could be no Dehalococcoides bacteria to do biological degradation of cis to vinyl chloride to ethene. And so I want you to consider whether cis stall is really stall, and whether you can get further reduction in degradation of the material by this material, magnetite. This is a test that Brian Looney taught me to do. We had samples from a site that was almost the color of chalk. It was white. And it doesn't look like it would have magnetite in it, but if you put a magnetic spin bar in the system and stir it up and let the magnetite accumulate on the spin bar and then pull it out, you can see that in fact it actually had a fairly substantial quantity of magnetite. If you've ever seen a child's toy where there's a face and then there's this black powder, and then you take a magnet and rearrange the powder to make mustaches and eyebrows and beards, that's magnetite. That's what it is. Commonly encountered in certain geological contexts. This is from a nice study done by the USGS on the distribution of surface lithology. Anything that's green was glaciated. And in our experience, it's relatively limited, but in our experience, outwash sands, particularly outwash sand aquifers in this glaciated environment, has enough magnetite in it to often matter for natural attenuation. So if your site is in that green portion, basically the north central and northeastern United States, it's worth looking for it. We've also seen it in material that forms from weathering of uplifted granitic or other igneous rock. And so certain parts of Southern California, the flank of the Sierra Nevadas, certain portions of the Rockies, and certain areas in the eastern United States associated with the core of the Appalachians, you have a reasonable chance that you'll have enough magnetite that it will be of some value or help. So let's do a case study for magnetite. I'm sorry, what I'm seeing and what's coming up is different. It'll take me a second to get to the next slide. Okay, here we go. Sorry for that. This was a site that Todd Wiedemeyer examined in some detail 20 years ago, and it was the site where Todd and I came up with the type 1, type 2, type 3 behavior processes. It was an old fire protection training pit at the former Plattsburgh Air Force Base. And so the A location there is just down gradient of the fire protection training pit. And this pit had chlorinated solvents in the water as well as BTEX compounds. And so locations A, B, and C are in the area where there was enough BTEX in the water to support biological reductive dechlorination. And then when the plume moved past the zone where BTEX was available, D, E, and F, then we saw a curious pattern of removal of the vinyl chloride and removal of the DCE that was not supported by the biological processes carried out by Dehalococcoides. So we put it up into BioChlor. And to explain the DCE removal, it required a rate constant of 0.2 per year. And then if you're going to use BioChlor, or for any model, if you're going to model up magnetite, it's important that the model does not mislead you. BioChlor is set up that any TCE that it removes, it produces that again as DCE. Any DCE that's removed shows up as vinyl chloride. But magnetite doesn't degrade DCE to vinyl chloride. So when you model the behavior of vinyl chloride, it's necessary to hide the DCE from the model. In other words, where you have input, you have to run the model twice, a second time for vinyl chloride. When you run it for vinyl chloride, just don't tell the model about the DCE that's at the site. Otherwise, you'll get a spurious production of vinyl chloride from DCE degradation. So when we do that, we get a rate constant for vinyl chloride removal of 0.4 per year. And I've spared you all the model iteration studies. So we have two rate constants now, one for DCE, one for vinyl chloride. We went out and took sediment samples for analysis of magnetic susceptibility. These were done by taking samples of the earth and taking them back to the laboratory and running the analysis on a simple magnetic susceptibility meter. It's actually fairly commonly found in geology departments, and it's used to understand and prospect for ferrous minerals or iron deposits. So we looked at 3 different locations and found that we got about the same number for magnetic susceptibility, which we're going to use as our predictor for magnetite concentration. So we put those numbers, the rate constants, and the value for magnetic susceptibility, and our spreadsheet, and we find that actually these rate constants are fairly slow. Most of our calibrating database has faster rate constants. And this is how it plots up. Again, the blue shape is expert judgment on the plausible range of values for magnetic susceptibility as a predictor of magnetite. and a first-order rate constant for degradation to DCE. It's right at the bottom, but there it is. We presented this figure at a number of conferences, and people look at that and they say, well, there's no correlation between magnetic susceptibility and the rate constant. Well, the reason for that is, for reasons that I still don't quite understand, the range of field samples that we've had access to are fairly constrained, a little over an order of magnitude wide. And the range of rate constants, again, are constrained. It's about an order of magnitude wide. So what this really says to me is that where we've been able to look, the magnetic susceptibility has been about the same, and the rate constant that we've derived from that has been about the same. Now, there's two kinds of rates in this plot that we're comparing to our field scale first order rate constant. The solid symbols are actually long-term incubation laboratory microcosm studies. So we take some sediment, we put some in a little glass bottle and incubate it for 2 years and see how fast stuff goes away. The solid symbols are microcosm studies. The open symbols are field scale rates that were done much as the way we did the Dehalococcoides study. A field scale rate constant compared to the field scale magnetic susceptibility. There's a number of interesting features in this. It doesn't seem to much matter whether it's DCE or TCE or vinyl chloride. There's no real difference in the observable rate constants. And there's no real difference in the field data and the laboratory data. I've been talking about this now for almost 10 years, and I really haven't understood why people haven't picked up on it and used it more. But I think one reason is nobody has any money left for site characterization, which means there's no more wells being drilled and there's no more core samples available for the analysis of magnetic susceptibility. So I wanted to take a look and see if we could also determine magnetic susceptibility using a downhole meter in a monitoring well that's cased with non-ferrous material like PVC. And I want to confess that I messed this one up. I talked some people into actually measuring magnetic susceptibility with a downhole meter for susceptibility, and I misunderstood the data that came out of the report, and I made a rushed to judgment that it didn't work because we were looking at the magnetic susceptibility of the filter pack. Well, I went back and looked at it, and what I had done is misinterpreted the units that were provided by the report of the downhole flow meter. This is a case study in a site in a deep, economically valuable municipal aquifer in Albuquerque, New Mexico. And this is the report off downhole Tooele logging of magnetic susceptibility. And it reported the values in the dimensionless units of volume magnetic susceptibility. And for these to be interpretable, these have to be converted to the mass magnetic susceptibility that I've used in the earlier figure. And basically what you do is divide by the bulk density of the material. And so you have to convert the volume magnetic susceptibility to mass magnetic susceptibility by dividing by bulk density in the units of kilograms per cubic meter. So I went back and looked at the reported volume magnetic susceptibility for 4 different locations. What those are actually average numbers over the full log. And then I took an average of the average, and then I divided by 2,750 or whatever it was, kilograms per cubic meter to get an equivalent mass magnetic susceptibility. I did not have the rate constant extracted from the field data using the model that I would have liked to have had. They simply didn't do that at this site. What I'm going to use is a rough surrogate, and it's the first order point rate constant for decrease in concentrations over time in selected monitoring wells. The source at this site is pretty well gone, and what we're looking at is degradation in the water. And so once the source is no longer contaminating groundwater, the rate constant at a point in the aquifer really is the rate constant of degradation in the groundwater. So as a first approximation, we're going to average up these point rate constants, get a grand average of about 15% per year, put that into our spreadsheet and plot it up. And it actually is pretty close to our blue shape, effectively indistinguishable. So this makes me have some confidence that it's worthwhile to actually acquire these magnetic susceptibility measurements two different ways. From core material if you have it, or by logging monitoring wells if the wells are screened in non-ferrous material. So we actually expanded the original M&A guidance that Todd Wiedemeyer and others created for EPA by including the benefit of these abiotic transformation processes. And we gave you a way of evaluating it in a quantitative way instead of just simply scoring it up and comparing it to scores, a non-quantitative pattern recognition approach. And we put all of this into a framework called BioPIC that'll walk you through the logic step by step and support that with more detailed information as you need it. I appreciate the chance to talk with you about this material. It's been very exciting for me to be able to work with this team and reorganize our new learning in a way that makes it accessible to people. And with that, Rula, I'll turn it back to you.
Thank you, John. We have a number of questions for you. Are you suggesting the use of this tool as another line of evidence to support M&A? And how do you convince regulators that degradation processes are sustainable?
The tool does provide the second line of evidence, that is its intention. The sustainability issue, as Carmen said, we didn't have enough data that we could actually go through and analyze a large number of sites and get some sort of general behavior. But if your regulator is concerned about the sustainability of the biological process, the Dehalococcoides reductive dechlorination process, then hand them the papers that came out of Heather Rictanus' work and Frank Chappell's work and convince them that you need to take core samples and evaluate the degradability of the organic matter in the system. Where people have bothered to do this, they'll find that in almost every case there's enough organic matter to sustain this process for centuries. But you have to invest in the site characterization, get the core samples, and go through the analyses that they did for dissolved inorganic carbon. Dissolved organic carbon. Dissolved organic carbon. For the process with magnetite, I'm working up some simple equations that will look at the erosion of the magnetite by the chlorinated solvents. If you have hundreds of milligrams, hundreds of thousands of micrograms per liter TCE, and decades of exposure, it's possible that you could burn the magnetite out. If you're looking at tens of milligrams per liter or less than 1 milligram per liter, it's probably not the case. But I haven't finished it. I'm still working on it, but I'm going to come up with some rules of thumb about sustainability for magnetite. The section
Thank you, John.
yes.
Please go ahead. I thought you were
Yeah, I'm sorry. One other comment. The section on iron sulfide also has an evaluation of the sustainability of the iron sulfide. So it's in there. Okay, that's all I have for that.
All right, thank you, John. Another question related to magnetic susceptibility. Can we assume that when we measure it, that we are underestimating the amount of reactive iron presence, since there are also non-magnetic forms of reactive iron that could be present in the subsurface?
All right, that's not a question, that's a statement of fact. And thank you for whoever sent that in. It only sees magnetic susceptibility. And at many sites we're going to have other reactive iron minerals, like the iron sulfides that I just discussed. Magnetic susceptibility does not see that. Like green rust, magnetic susceptibility does not see that. And like certain clay minerals that have iron too as part of the structure of the clay. And so magnetic susceptibility only sees one portion, though I think it may be an important portion at some sites, of the total abiotic processes.
Thank you, John. Is BioPIC usable for trichloroethane for TCA releases also?
Not really. It wasn't designed for it. You'd have to have some expert knowledge to work your way through BioPIC, but really I think what we need to do is somebody needs to work up a BioPIC for the chlorinated alkanes. But that
right.
wasn't part of the original charge for Carmen's framework study.
Thank you. And can you tell us if stable isotope analyses have a role in the new framework, and if so, where it all fits in?
They do have a role. One of the important questions is vinyl chloride, DCE, PCE, TCE degrading? Is it degrading? And there's actually a spreadsheet that plots up stable isotope values, the delta 13C value, and information on the reductions of concentration to see if you can attribute or recognize, not attribute, but recognize degradation of the compounds from stable isotope fractionation. And so if you have that kind of information, then there's a tool that allows you to determine if it shows that the compound is degrading. And if you really feel like you need to support that it's degrading, if the results of the model are a little bit inconclusive or a little bit problematic, then you can use the stable isotope analysis as another line of evidence to document it, that it's degrading, or document if it's degrading. We did not use it as a line of evidence to predict the rate constant. We've wanted to do that. We've tried to do that successfully at several sites, but most of the time at most sites, the reduction in concentrations are a combination of processes, including dilution and dispersion. And so the formulas and calculations using isotopic fractionation fail to identify the rate constant for degradation. But it's a very useful, very viable, very powerful tool to determine qualitatively whether degradation occurs or does not occur.
Thank you, John. One question related to rates. Do you have to confirm plume stability before calculating degradation rate?
I'm quiet because I'm thinking about it. I'm going to inflict a diversion to this questioner before I answer his question. Plume stability, as defined by the OSWER 98 directive, relates to whether a plume is moving forward in three-dimensional space along a flow path. Stability is defined in terms of the geology of the site and where the plume is encountered. People have taken an approach of working up monitoring data for wells and looking at trends and concentrations and saying that that tells you something about stability or not about stability. And that's actually, in my mind, an abuse of the statistics. The statistics can tell you if something is happening. You cannot tell you if something is not happening. So stability then is a property of the behavior of the plume in three-dimensional space. Now, does the plume have to be stable? Actually, no. If the model is implemented as we show it, it does allow a possibility for a plume to expand. The question is, when it's finished expanding, will it reach or will it not reach the receptor? So no, you don't have to reach plume stability to apply the protocol.
Thank you, John. I have a question for both you and Carmen, and I'd like to open it up and have Carmen maybe start with the answer and have you join in.
I'm here.
Great. How can BioPIC be obtained and when?
Actually, Deanne sent it out as a reminder this morning. It was a reminder for this call. It has two files attached. One is a spreadsheet, which is the BioPIC, and the other one is a PDF file, and it's just a quick guide on how to use it. But if you didn't get that, feel free to email either me or John or Todd or Frank.
Great. Thank you, Carmen. And Carmen's contact information as well as John's contact information is included in a PDF of these slides, which you can download from the Startup and ESTCP webinar series webpage. All right, so question for both of you again. How will the framework and tool be updated as new methodologies emerge or as the science gets updated?
Wow. Coincidentally, both John and myself retired within this project, and this is something that we have often wondered about. And I'll leave this to John mostly because I think at this point we have captured most of the processes that capture reductive dechlorination, and it'll be a while before something comes up, but I'll leave that to John.
Well, there are some things that we're just not quite ready. For example, trying to understand aerobic cometabolic degradation of TCE by oxygenase enzymes. We just simply didn't have the rate constants that could be associated with the predictors of the rate. So it's not done. You have to understand that this ESTCP project is a product of a government agency, and it's required to go through a review process, and then it's only released once it's been modified and edited to respond to the reviewers' comments and questions. So once it comes out from ESTCP, that's what the product is. It won't be changed until there's a follow-on project. However, I have been talking with Carmen and Todd about informally updating it. But again, informal means that it's only John and Todd and Carmen, and that the ESTCP has no responsibility to it or for it. Be interested to see if people think that that's a valuable contribution or not. But the short answer is the ESTC project will be fixed in time once it's gone through review and clearance.
Thank you both. And one last question. What Superfund decision framework do you recommend using, and how can BioPIC be utilized to augment this decision framework?
I don't understand the question really, because there's only one Superfund decision framework, or there should only be one. However, many Superfund sites don't fit their own framework because they have not identified a time by which the cleanup has to be attained in the record of decision. But the existing BioPIC framework really is designed for a RCRA type system where you expect to continue to use the facility opportunities for contamination will continue, and that you're required by the regulators to basically maintain and control your contamination, but not necessarily clean it up. And cleaning it up is another question that this is not appropriate for.
All right. Well, thank you, John, and thank you, Carmen. And on behalf of SERDP and ESTCP, I'd like to also thank all of you for attending today's webinar. As a reminder, the presentation and audio will be archived for future reference on the SERDP and ESTCP webpage. Our next webinar is on Thursday, March 26th, and it will focus on innovative tools for species inventory, monitoring, and management. This webinar will feature two speakers, Dr. Karen Goldberg from Washington State University and Dr. Lisette Waits from the University of Idaho. And you can register for this free webinar using the link shown on these slides. So this concludes today's webcast. When you close your window, you're going to get a survey. Please take a moment of your busy day to complete this survey, as it will help us to make these webinars better in the future. So thank you all again, and a reminder to go ahead and download the PDF and download the information, the tool, and the tutorial on the tool in the links that were sent to you. Thanks, everyone.